talking about the digestive system. We were on our tour as we were going down the system, and had gotten to the stomach. And we had been talking about the fact that a lot of people focus on the stomach as kind of the most important organ of the digestive system, and it certainly is important. It has various roles, like all of our other sections of the alimentary canal and our accessory organs, etc. But we were suggesting actually that the section that comes after this may be even more important. So what comes after the stomach if we're going along the alimentary canal? Small intestine, exactly right. So we will find that the small intestine has 3 different sections to it. We can see just the first little section right there, called the duodenum or duodenum, depending on where you're from. And then that's going to go into the jejunum, and then the ileum finally, before that comes into the large intestine. So we had gone through some of the anatomy of the small intestine, like we see there, and we're going to start talking about a little bit more of the physiology. We had talked about the fact that there were numerous secretions that come out of the stomach. So because there's a lot of mucus, because it's going to be along this path of the tube that opens to the outside, so it's always going to have a mucus lining. The mucus lining is a little bit thicker, a little bit alkaline actually in the stomach, and it helps to protect the stomach wall from kind of being eaten away by the stomach acid, the hydrochloric acid, etc. And that's another secretion that we see coming here from the parietal cells. And then we also have a secretion coming from the cheek cells. So this is pepsin. When it gets originally secreted, it's called pepsinogen, which is like fibrinogen when it's in an inactive form. But for the pepsin, in order to become activated, all it has to do is go into a low pH environment like that in the stomach with the hydrochloric acid, and then it's going to be able to do its job in starting to chemically digest proteins. So again, at the very end of this chapter, we'll talk a little bit about nutrition, about the different molecules that we're looking to break down, and we've been kind of highlighting that through time. So for instance, protein, the protein is going to be what's called a polymer. Poly means many. So with a polymer, that's going to be the big form of the molecule, and then when we start chopping it apart, we're going to chop it into shorter strings of amino acids. Those shorter strings are going to be called peptides, so P-E-P-T-I-D-E-S. So proteins get broken down into peptides, and that's one of the first things that we're going to see happening chemically here in the stomach. So proteins are going to get broken down to peptides. Now that's not as small as it needs to be. With those peptides, those strings of amino acids, we need to keep cutting those up to get them down into individual amino acids, or even maybe 2 of them maybe linked together. So we need to get them very small in small packages like that down to what we call the monomer form. So mono means 1. We want to get it down to the single unit form as opposed to the multi-unit form, polymer down to monomer. So, so this is the first step in the process. This is the first chemical digestion of protein we're seeing in the stomach. Now we did see another organic molecule starting its chemical digestion up in the mouth with salivary amylase, looking to digest what? Yeah, starch like carbohydrates and starches, right? Yeah. So chemical digestion of carbohydrates started up in the mouth. Now we see the chemical digestion of protein starting in the stomach, and we still haven't seen fats being digested yet, or lipids, so we're waiting to see that. Okay, so where we stopped then is we're just about to talk a little bit about the coordination of the activity of these different digestive organs. So we said that it's a bit like a disassembly line. So in each place along the way, each one of these organs, accessory organs, or even ones along the main street, have a particular role to play, like we've been seeing so far. And for that disassembly line to be effective, we want to make sure that if this section right here is slowed down for some reason, that the other sections are going to be aware of it so that they can coordinate their activity appropriately. So if this one's slowed down, then obviously the section before it needs to slow down too. So we don't want to just keep piling food into the section that's moving slowly for whatever reason. So we're going to find that our 2 different control systems of the body are going to be very well used in coordinating the activity of the digestive system. So what are our 2 different control and coordinating systems of the body that we've talked about already? Like whose? What? So sympathetic and parasympathetic sympathetic nervous system. There are the autonomic ranges of the nervous system. That's very good. So those are going to be coming into play in terms of increasing or decreasing the activity of our different organs, like the digestive, right? Parasympathetic branch is the phrase that goes with it is rest and digest. And so for the digestive system, parasympathetic stimulation is actually going to increase the activity. It's going to increase the mobility, the contraction. It's going to increase the secretion and all the different activities we're talking about. Sympathetic, on the other hand, is going to slow the activities of the digestive system down because we don't want to spend energy on that type of thing if we're stressed out. The other control system that we have, besides the wired nervous system, is the wireless endocrine system. So endocrine system, right, they'll use chemical messages to communicate to different distant target cells, and that's certainly true here as well. So we will find that there's a number of hormones that will increase or decrease the activity of some of these different digestive organs. So for instance, there is a hormone that is called gastrin, that comes from the stomach that actually increases the activity of the stomach. So it's almost like self-stimulating. And you look at the word, right, it says gastrin, like gastric, reminds us of stomach. And so this is secreted by a certain subset of cells in the stomach lining, and it tells the parietal cells to increase their secretions, and the chief cells to increase their secretions, etc. So you can imagine when food arrives there, and stress receptors go off, or there's chemoreceptors there, and says, hey, we just, got a bunch of meat because you're eating some chicken or something like that, that's going to especially tell the stomach, or make, in essence, increase the stomach activity because one of its main roles is to help digest proteins. Okay? So gastrin is an example of a hormone that increases our activity in the stomach, particularly the secretions, like we were talking about before. But then other types of chemicals will tell us to slow down. So for instance, this one right here is called cholecystokinin, and that one is oftentimes abbreviated CCK, which people like much better. And CCK comes from the intestine, and what it does is it actually goes back and inhibits the stomach activity. And to a certain extent that makes sense, because if a load of food gets down into the intestine, it's got a job to do. It's going to be busy for a while. So it will want to communicate back upstream and say, stomach, you can slow down for a while because I'm working on this. Don't send me any more stuff for a while. Okay? Once I empty this out, once I kind of mix it around with enzymes and send it on down further, then I'll be ready for whatever you have. Okay? So this gives us the idea of at least a couple hormones which can increase or decrease the activity of the stomach. Now this one right here is an example of the nervous system control of the stomach, like we talked about the parasympathetic stimulation to the stomach that can increase its activity. And so it comes down and tells the stomach to increase its activity. It's going to increase its secretion, then it's smooth muscle contraction and things like that. And all of that as well may have an effect also on the hormonal control, like we talked about in terms of gastrin, to make sure that the stomach is controlled well so that we can make sure that it's digesting the food as appropriate. So when we're talking about the digestion that's going on in the stomach, though, like we were talking about before, this is a very long path. And right now, we've only gone maybe probably less than a third or a quarter of the way through it. So you can imagine that much like an assembly line, right? If you've only got a third of the way through the assembly line, then you don't have a finished product yet, right? It's probably only a third assembled. And if you want to think of it in terms of our disassembly line, then our food items are probably only, a quarter or a third disassembled or digested at this point. So it's not appropriate for us to try to pull things across the body wall because things are still too big for us to be pulling into cells, lining the tract, etc. So there's very few things that actually get absorbed in the stomach. So small quantities of water, right? We don't have to break that down. So you can absorb small quantities of water, certain salts, and those will certainly be tied together, right? When we move sodium across the membrane, right? Usually it sucks the water with it through osmosis, etc. A little bit of alcohol can get absorbed in the stomach, and some lipid-soluble drugs like aspirin, etc. But even those types of things we see there, that we can get some small absorption of those chemicals or those ions or whatever, still, if we compare the amount that we might absorb in the stomach at one time compared to what we're going to get downstream of the small intestine, it's relatively small. Okay? So while for some of those things that are already, that we don't have to digest really, we can pull them across to the stomach, a lot more of it is going to be absorbed in the small intestine next. Okay, so what's going to happen here is that the food is going to arrive in the stomach down from the esophagus, and we're going to be mixing it with a lot of gastric secretions. So it's mucus, and pepsin, and hydrochloric acid, etc. And so what we have there is this kind of acidic soup that we call chyme. You see the word right there, C-H-Y-M-E. So chyme is this acidic soup that's in the stomach. So you can imagine with a lot of fluids and stuff like in there, you're going to want to spend time digesting that food. You're going to want to absorb a lot of nutrients and fluids from that food before we get rid of it. Okay, so that acidic soup, that chyme, is kind of analyzed. We have different receptors that will analyze it in terms of how concentrated it is, how much fluid is there, what's inside of it, etc. And so if it's high protein content and not very fluid, then it needs to spend some more time in the stomach. We need to put more secretions with it. We need to mix it around and churn in the stomach a bit more so that the stomach pepsin and the hydrochloric acid have a chance to break those proteins down a bit more. But if it's pretty fluid, we've done a lot of secretions, and maybe it doesn't have a lot of protein, like we ate a pretty balanced meal so that there's not a lot of protein in it, then once we've done our job of adding mucus to it and hydrochloric acid, it's time to send it down. Okay, so it depends on what it is and how much there is as well as to when it moves to the next section. Okay, so we were just coming from this area up here. Okay, so this is where the stomach was located, right? So if we were to draw it, we would see that the stomach is going to be our J-shaped organ that was extending up this way like that. Okay, so it's coming down there. We see the pyloric sphincter, right? That band of smooth muscle right there that is going to control the flow between the stomach down into the small intestine. And usually it's like maybe a teaspoon or a tablespoon at a time that will actually be let go into the small intestine. So just a little bit at a time. A lot of times that pyloric sphincter is closed, and that way, when the muscular contractions occur, it'll squeeze that acidic chyme down to this kind of closed pyloric area, and it'll be like a washing machine. So, or like if you ever see like the waves come in against the shoreline, rocky shoreline, and it will kind of splash up against the rock and then come back, and then splash up against the rock and then come back. That's a bit about what happens down in that narrow portion of the stomach down towards the end where it transitions into the small intestine. If that pyloric sphincter is closed, we're going to get a lot of mixing and churning that's going to go on in there. Okay? Okay. So once we are ready to go to the next section, then we're going to be let through that pyloric sphincter, and we're going to go down here into the first section of the small intestine, which we call the duodenum. Now the duodenum is the shortest of the three sections of the small intestine. It's only about 12 to 16 inches long, right? So it's about a foot long out of a number of meters, right? So we said it was 27 feet long, and so the small intestine is the bulk of that. So 12 inches is a relatively short section of the small intestine. So when we look at it, it's again going to be a place where we're going to do a lot of mixing because we haven't had a chance to do a lot of digesting yet. And we've got three accessory organs that will dump their secretions in there. We've got the feather-shaped pancreas that is deep to the stomach. We've got the green teardrop-shaped gallbladder. And then we've got the liver, the big triangular-shaped liver that sits on top of the gallbladder. So if we were to get that in there and add a big liver, that would be, in essence, kind of up here, like this. Okay? So all three of those organs are going to dump in right there at the duodenum. And there is a section right here. Let me get a better color. There's a little section right there, a little papilla, a little bump, where all those secretions dump in. So we've got right here something that's called the common bile duct, which is bringing bile down from the gallbladder and from the liver, like you see here. Okay? And then also, we've got what's called a pancreatic duct, which is bringing in secretions from the pancreas, like we see here. Okay? So all three of those organs are going to secrete something that's going to be very important to mix with the food to aid the digestion of it. So that duodenum is a big, again, mixing center. So it's an area where all these secretions are going to come in from these other accessory organs. So let's talk about these accessory organs. We'll start with the feather-shaped pancreas. And the pancreas is pretty darn important. Now, it puts out what's called pancreatic juice. That's pretty straightforward. Okay? Now, we talked about the pancreas once before when we were in the endocrine system. And we said that the pancreas, we focused on two hormones that the pancreas puts out. You guys remember what those were? Diabetics and stuff like that have issues with, insulin and glucagon. So those work to regulate the amount of glucose that is circulating around in the blood. So after we eat a big meal, we've got a lot of free glucose circulating around. We want to store it for later. And so then the insulin is going to cause that glucose to be stored in the liver and in the skeletal muscles as glycogen. Glucagon has the opposite effect. It's going to cause that glucose to be gone from the cells, put it back in the blood so that we have it available. Anyway, that feather-shaped organ then is dual function. It's partly endocrine and it's partly here digestive. But when we say partly, it's not 50-50. Okay? There's probably about 1% of the pancreas that are these little pancreatic islets that actually put out the insulin and the glucagon that we talked about before. 99% of the pancreas is going to be made up of these things that are called the feeder cells, and these are going to be the cells that are putting out pancreatic juice. Now, pancreatic juice is a bit like gastric juice, right? There's going to be, in essence, some liquid to it, and then there's also going to be enzymes that are coming out, and they're going to dump into the duodenum. Okay? So that pancreatic juice is going to drain to the pancreatic duct like we saw before. There's a little span of smooth muscle. It's called a hepatopancreatic sphincter, right? Anytime you see hepa or hepato, that refers to liver. And we saw those two ducts coming down to that one place, ducts. And these are ducts, not quite by accident. It's D-U-C-T, ducts, tubes, coming down from the gallbladder and the liver, right? So that's the hepatic part. And then we have the pancreatic duct coming across, and they're bound together. So that smooth muscle band controls how much of these secretions will be dumped into the duodenum. Okay, so what is the pancreatic juice? Well, there is some sodium bicarbonate. So sodium bicarbonate is very important because what's coming from the stomach is that gastric juice. Gastric juice, we said, is like an acidic soup. And so if there's an acidic soup coming down into the duodenum, there's a possibility that acid, as you might think, could eat through the wall of the duodenum. Now, the stomach has various ways to protect itself. It's got cells that replace themselves very quickly. They're kind of sealed off from the hydrogen ions, et cetera, et cetera. Duodenum doesn't have those protections. So we've got to neutralize that hydrochloric acid that's coming down to the duodenum. And so that's one of the roles of the pancreas, is it's going to put out the sodium bicarbonate. But in addition to that, the pancreas is going to put out a whole buffet of enzymes to digest the different food items, organic food molecules that you're taking in. So remember, those are going to be carbohydrates and proteins and lipids, and then technically also nucleic acids, right? Because if our cells are going to be making new cells with RNA and DNA, we've got to have those subunits, right? We've got to have the nucleotides, the little building unit, in order to make that entire sequence, to make genes, to make RNA, like messenger RNA and all that type of thing. So we've got to have those subunits coming from somewhere. It's going to come from the nuclei or the cells, the animal plant cells that we take in. So we've got to break those down too. Okay? So we've got enzymes for every single different food molecule that you're going to take in to assist in the chemical breakdown of them. And if we look at the names, we can probably figure out what these enzymes are going to break down. Okay? So pancreatic amylase. We saw amylase already up in the salivary gland. So salivary amylase broke down the starch that we talked about before, carbohydrates. So this is our second round of carbohydrates. So we had a big, long polysaccharides. We started chopping them up, but we need to get them down to like monosaccharides and disaccharides, those simple sugars that you hear about, like glucose and fructose and maltose, etc. Okay? Even lactose, right? So that's milk sugar. Okay, so pancreatic amylase is going to work on finishing that chemical digestion of carbohydrates. pancreatic lipase. What do you think that's going to break down? Yeah, exactly right. So lipase looks like lipids. Anytime we see this ASE, that indicates that it's an enzyme. Okay? Now, not all enzymes will have ASE suffix on the end, but if they do, if you see that H on the end, it indicates that it's an enzyme. So this is an enzyme that helps to break down lipids. Okay? Then we have a few that work on proteins as well as peptides. So, for instance, carboxypeptidase, you can see the peptid part looks like peptide. There's the ACE part at the end. So what we're doing is we're finishing chopping up the proteins with those enzymes as well. So we're going to get those down to individual amino acids, and then there's two different nucleases, one for the deoxyribonucleic acid and one for the ribonucleic acids. Okay? So that'll be important because when we get to the small intestine, we said that not only is this a major site of digestion because of all these enzymes that are coming in from the pancreas, but it's also the major site of absorption. So if we're going to be absorbing it in the small intestine, that means it finally has to be broken down small enough that it's something we can pull into those cells and put into the blood, etc. Okay? So this is it. We've got this. No more, oh, we're going to digest protein for a little while. Oh, we're going to digest just carbohydrates for a little while. Now we need to digest everything and get it broken down. Okay? So the pancreas has a very important job there. Now, just like the stomach, the pancreas is going to be controlled both by the nervous system as well as by the endocrine system. So there's different types of hormones that will affect its activity. So for instance, there's a hormone called secretin that will, as you would imagine, increase its secretions of its different types of pancreatic juice. So for instance, if we see here, cytokines, there's like CHYMB, cytokines coming into the duodenum. There are going to be chemoreceptors, right, that can detect the pH changes there. And what's going to happen is secretin is going to be released by that duodenum. It's going to go up, and it's going to stimulate the pancreas saying, hey, pancreas, we need a lot of that sodium bicarbonate. So those bicarbonate ions can bind to the hydrogen ions and neutralize them, just like we talked about in the respiratory system. Okay? So the bicarbonate ions are going to neutralize the acidic ions, and it's going to help to protect the lining of the small intestine. Now this one, cholecystokinin, we already mentioned, right? This is that CCK. Again, it has an effect on the stomach because the stomach can slow down, but when this is released by the small intestine, it's telling all the accessory organs of the small intestine, hey, I got a load of food here. I need your secretion to help me digest it. So CCK is going to tell the pancreas to release its various enzymes. Okay? So we've got two hormones that we can use to communicate from the small intestine to the pancreas. As well, these hormones will have an effect upon the gallbladder and the liver to make sure that the small intestine is getting all the components it needs to help break down the food that's arrived there. Okay, so that's the pancreas. The pancreas puts out sodium bicarbonate to help neutralize the acidic acid, and it puts out enzymes to help digest all our different food molecules. So now we go to the liver and the gallbladder. And the liver and the gallbladder are going to be tied together in terms of their function. So the liver is the largest internal organ of the body. What's the largest organ of the body you always hear about? Yeah, the very first system that we addressed, that's the largest organ of the body, because it counts as an organ system and an organ, etc. Internal organ, the liver is the largest one. It's a big triangularly shaped organ, has different lobules in it. This is a big zoomed up view of the cells inside of the liver. So each of these cells that you see here is called a hepatic cell. So remember, anything you see hepa and hepato means liver. So these are often called hepatocytes, right? Now what we'll find is that this whole thing, this big kind of brown thing that you see here, this is called liver lobule, and the cells are kind of lined in almost like spokes on a wheel. You can see that there's a central vein right in here that's, according to my drawing, not very central, but it does. So, the liver has a ton of different functions, So it makes like plasma proteins like we talked about before. It gets rid of toxins that are in the blood. We know it's a place where red blood cells will go and die in addition to the spleen. There's lots of different functions. It's got like 30 plus different functions, okay? It's got amazing powers of regeneration. You can donate a lobe of your liver to somebody else, and in essence, your lobe you're missing will grow back, and they can grow almost an entire liver from the lobe that you gave them. There's very few organs that do that. Right? You can't give like a chamber of a heart to somebody, and your chamber grows back, and they just end up fine too. Same thing with the lung, etc. But in any case, what we want to focus on in terms of the liver is what does it have to do with digestion? How does it benefit us in breaking these food items down? Okay? So here's the liver, like I said, triangular shape. So it's mostly on the right side, but it's big enough that, if this is the side of your body right there, the other side of your body is probably right about here. Okay? It almost goes to the entire left side as well. But the main part of it, the main big bright lobes on the right side of the body, and we can see that the pancreas, not the pancreas, but the gallbladder is partially hidden by it, right? So the gallbladder is just under there like that. So we see it flipped up over on the right-hand side there. And we said that the gallbladder and the liver have functions that are tied together. Okay, so for instance, we said that the liver has a lot of different functions, so it does a lot of processing of different food items that we've broken down. It does a lot of storage, like we said, there's like a glycogen molecule we said for glucose, different vitamins, blood cells are going to kind of be recycled there, things like that. But the biggest role in terms of the digestive system is right here. Okay? The liver produces bile. So, the liver is the organ that makes this green substance that's called bile. Now, we just got done with another organ that looks very green as well. What's that called? Gallbladder, right? So, the bile itself is made in the liver, but it's stored in the gallbladder. Okay? So that's the tie between the function of the liver and the gallbladder. Is, like for instance, when you have food going down to the small intestine, well, the liver and the gallbladder are going to release bile. The liver is going to release some freshly made bile because that's its role in the digestive system is to make bile. But the gallbladder is going to release the bulk of it because, kind of throughout the day, in between meals, the liver can kind of slowly go, make bile, and then just store it into the gallbladder. And then now that we have a meal come through, the liver doesn't have to panic and produce all this bile mixed in with all the food you've taken in. It can just release most of it from where it's been storing it into the gallbladder. Okay? Okay, so like I said, bile itself is kind of a yellow-green liquid that's created by the hepatic cells, the liver cells, and it's got lots of stuff in it. It's got water, right? So it's a liquid. It's got bile salts, pigments, cholesterol, and electrolytes in it. But the bile salts are what's really important for the digestive system. So what it does, and we'll use this figure or this slide and have some space in it, right? So it's going to be made in the liver, it's going to be stored in the gallbladder. And say we've got a bunch of different fat droplets like this, okay? So when fat arrives down to the small intestine, fat is kind of a hard molecule that works to break down. It's slippery and stuff like that, like oil and butter. It's the same thing inside of the body. And so we have to deal with it in a special way. Now, we already said that we've got an enzyme coming from the pancreas that helps us to break down lipids or fat. So what was that called? Has an H on the end, break down lipids. Exactly, right. Yeah, we're together. It's called lipase. And so lipase is the enzyme that's going to help to chemically chop up our fat molecule. And usually what we have is we will have, for instance, we'll have our triglycerides that you hear about. Okay? And we're going to break those down into monoglycerides and fatty acid chains. Okay? So you can see triglyceride 3, monoglyceride 1. So if you were to kind of draw it, you might have something like this. Okay? There's going to be a glycerol molecule that works as kind of the backbone, and then you have 3 fatty acid chains that are attached to it like that. So that's kind of what the molecule looks like. That's a triglyceride. So if we were going to break that apart, we're going to chop these off right there. So then it gives us the monoglyceride backbone, like you see there, the glycerol, and then the free fatty acid chain. Okay, so we have the enzyme coming from the pancreas called lipase. But when fat gets arrived down the small intestine, it tends to hold together, right? So if you guys ever seen like oil and vinegar dressing or like thyme dressing or something like that, before you shake it up, you've got these water-soluble parts like the vinegar and stuff like that, and you have the oil, right? They're separate. And then in order, before you put it on your stomach, you have to shake it up. And then physically kind of breaks up the large fat globules and makes them smaller so you kind of mixes it, right? But there's also a chemical way to do that same thing, to make sure that some of our different kind of water-soluble components can get in amongst the fat molecules to aid in their digestion. So what I drew over the right-hand side here represents some bigger kind of fat droplets, right? So like if you were washing a greasy pan or something like that, you make some sort of casserole, sometimes you're soaking in the sink and you see those kind of orange fat droplets sitting on top. If you take soap or something like that, like your dish soap, and you kind of drop some in, you'll see that oil sitting on top of the water, it kind of runs away, like totally separated, it runs away from wherever the soap was in. And what is happening there is the soap works as what's called an emulsifier. So let's write that on there as well. So emulsify or emulsification. Okay? So emulsification, what it does is it breaks up big fat droplets into smaller ones. It just kind of breaks apart those bigger ones. And if we can break those apart, then what we can do, right, so instead of having like those three bigger ones, if we can break them apart into all these little ones like this, then what can happen is that our enzymes that we talked about before, like lipase, can get access to more areas of the fat molecules, of the triglycerides, etc., so that they can actually get in there and chemically digest those triglycerides, okay? If we don't have the bile, that's what the bile does, right? It acts like soap. It works as an emulsifier, breaking apart these large fat droplets into much smaller ones so that the lipase can get in there and do its job, okay? So if we just have it like this, then you can see the lipase can get to the surface of it here, but there's no way to gain access to all the different fat molecules that are on the inside there. It can just eat away at the surface and that's it. So you would have oftentimes fat that would be undigested that would be passing through. So if you have lipase enzyme, it's going to digest fat, but it's not going to do it completely because it can't get access to it all. But if you have bile plus lipase, then it's going to work very, very well. So it's a bit like, I don't know, like if you're doing laundry or something like that, right? So if you're doing laundry and you put your clothes in there and you just kind of left it in there, you put the soap on top, and you just kind of left it in there, soap, right? It's not going to get in amongst all your clothes. The whole, detergent action there is we're going to need to break apart those clothes so the soap and water can get in amongst them so it can get clean, right? So the same thing here. We need to break up those big fat droplets into smaller ones so the enzymes can get in there and do your job. Okay? So that's the role of bile. It acts as an emulsifier, a bit like soap, in order to break up these bigger fat droplets like you see here and break them up into smaller ones and let the lipase, the enzyme, do its job. Okay, so bile comes from both the gallbladder and the liver, and its control is going to be, again, here you see, cholecystokinin, right? Cholecystokinin will stimulate primarily the gallbladder because that's where we have it stored to contract and to move muscle along the wall, and that will squeeze the bile down the tube and into the duodenum so it can mix with the food. Okay? So you can imagine, if it's an especially fatty meal, you're going to need a lot of bile coming from the gallbladder, a lot of that pancreatic lipase, etc. Now sometimes you hear about people who've had gallstones, etc. So the cholesterol that's part of bile can actually accumulate and form solids as opposed to a liquid. And if that happens, what do they end up doing usually? So they'll actually, and you can have solids removed, but oftentimes they'll actually remove the whole gallbladder. So they'll just cut it out, take take it out, because if you're accumulating gallstones, they don't have a good way of preventing you from accumulating gallstones. Just going to keep doing it and doing it. And like kidney stones, you can imagine this is a tube system. And so if you have this hard thing, much like a soft like an embolism or something like that, it blocks the tube and everything is backed up. It increases pressure. So kidney stones, right, blocking the tubes like yours and all of the kidney backed up, etc. Same thing with the gallbladder. So if they take the gallbladder out, okay, we can still have bile, because the bile is not created in the gallbladder, it's created in the liver, right? So you can still have bile being made, but it's much harder on the liver, and the liver can't really do it as fast as you would normally ingest as food, especially then if you are eating like fatty meals, etc. So those people that don't have a gallbladder, they have to watch what they eat, because if they eat a high-fat meal, then it's just going to be bad. They're not going to be able to digest those fats enough. And so they're not going to absorb them enough, and so then, whoop, it's going to be coming out the other end, not digested. So it's kind of like that whole Lester thing, which prevents you from absorbing fat, right? Okay, so you can live without your gallbladder, but the low bit of stress on the system. Alright, so that's where we see this kind of feedback loop here. This is, like I said, where cholecystokinin is going to stimulate the gallbladder to contract when we have that food arriving here in the duodenum, especially if it's high fat enriched food. Okay? Okay, so that's what we did. It's bile salts. These are one component of the bile. Emulsify fat into smaller droplets, so that we can break them down so that we can absorb these certain byproducts from fat that we ingest. Okay. So we arrive at the duodenum. But the funny thing is, when we're talking about the duodenum, we spend our time talking about the intestine organs that are all up in there. Liver, and gallbladder, and pancreas. So now we need to get back to talking about the small intestine itself. Okay? So we said that we have three different parts to it, and this is where we're going to have most of our digestion and our absorption occurring. So if it was the first part we were talking about the duodenum, then the jejunum, and then the ileum. Now in the small intestine, remember a long time ago we were looking at the wall of the ileum and figuring out, because it says the wall has a same four layers to it. We've got mucosa on the inside, we've got a submucosa where we have the glands originating, then we have a muscularis layer where all the smooth muscle is, then we have a serosa on the outside. But each section of it is a little bit different depending on the function of that section. So for instance, the stomach, instead of having two muscle layers, a circular and a longitudinal, it has a third muscle layer called the oblique layer. Because there's a lot of surgery that goes on in the stomach. It's a big organ. We got to make sure that we're going to be able to move stuff around. When we get to the small intestine, the wall kind of changes again. Now, in the stomach, we saw some wrinkles called rugae, R-U-G-A-E, in the stomach that allow it to stretch out. It's kind of like when you see dehydrated grape, right? Dehydrated grape is raised, right? So, you're seeing that same surface area of skin of the grape, but as the food in the inside of the grape raw, it can shrink, so you get those wrinkles. Same thing is true to the stomach. Those wrinkles allow the stomach to, when it's full, stretch out. But what we're going to find when we get down to the small intestine is that number one, we're going to have a lot of surface area. So we start out with a short duodenum, then we have a long jejunum that then transitions into the last section, which is the ileum, like we saw there. So it's a really long part of the alimentary canal, which means that the food is going to spend a long time in it, right? Because that's moved through this section. So number one is long. And it's got this connective tissue that's called mesentery that's suspended inside the cavity like that, allows the blood vessels, lymphatic, and nerves to get into it. But the other thing that we see about it, when we look at it, and I'll come back to those, is that the inside of it has all of its own little wrinkles to it. Number one, it's got these folds in it called circular folds. So circular folds go around on the inside of it. And so we have folds in it, just like a bit like we saw in the rugae, right? Of the stomach, but we have circular folds here. And on those circular folds, we have these little finger-like extensions like you see here. villi are plural. So we have little finger-like extensions of the mucosa that are going to extend off these circular folds. So each one of these little big folds and finger-like extensions gives us more surface area, more places where we can secrete stuff onto the food, where we can absorb stuff from the food once we finally broke it down, etc. So not only is this long and long, but it's got all these different levels of increasing surface area. We have more area to be able to deal with this food and get it kind of finely broken down and absorbed. Now, you would think that with these finger-like extensions, these little villi like we see here, that would probably be it in terms of these little adaptations for surface area. But when we look at this, right, along here, when we mucosa, we have these tall epithelial cells like this, okay? These tall epithelial cells are a single layer cells. It's simple columnar epithelium. And when we have these simple columnar epithelial cells, so say here's your cell, like this, your nucleus down to the bottom like this. On the apical surface, the surface that's facing where the food is, it has its own set of villi on each and every cell. And these are called microvilli. And that's what was on slide 4 we see here. Okay? So each and every columnar epithelial cell that we see, like right here, each one of those has a microvilli on its cell surface. So again, now we have this whole other layer of increasing surface area in order to create more enzymes, in order to absorb the food, etc. Okay? So there's lots of different ways that we have of increasing the amount of surface area we have inside of the intestines. So we have the villi, like you mentioned, and inside the villi, like we saw in that picture. Remember we were talking about the lymphatic system, right? So here's the lymphatic vessel, and look, there's one lymphatic vessel that goes right up to the core of each villus, okay? That's called the lacteal. We said that the third function of the lymphatic system after fluid collection and bodily defense was to help treat quarterbacks. Okay? So each one of those each and every one of those villi, we have a lacteal lymphatic vessel that can help you down the back. Okay. So this little set of microvilli we see here is often called brush border because it looks like, if you look at it in the microscope, it looks like a little brush or something like that that comes along the surface of those cells. And it is right there where we have more embedded digestive enzymes. So not only does we have all these enzymes released from the pancreas, right, for lipase, and the different proteolytic enzymes, etc., but now we're seeing more enzymes, right? These are the last step, right? Peptidases. So peptidase that tells us that it's an enzyme. Peptidase looks like what? I don't know. Looks like peptidase, I don't know. Peptide looks like peptide. Peptide, we said, is the second kind of step of breaking down proteins. So remember proteins, we start out with large proteins like this, and then those break down into shorter chains that are called peptides, and then from there we get down to the individual building blocks of the amino acids. Okay? So we start breaking down proteins into some with our peptide. Now we need to break down these peptides into smaller units down to the amino acid level. Now also when we look at enzymes here, we find that, right, we have enzyme sucrase, that's for sucrose, right, one of our simple sugars. Maltase, that's for maltose, another simple sugar. And lactase, that's for lactose, a milk sugar. Okay? Those people that are lactose intolerant do not make this enzyme lactase. And so they can't fully break down the product in the sugars in milk. And so anytime, like we're not trying with fat or something like that, if you don't break it down, if you don't get it small enough, you can't absorb it across. And that means it's going to continue to progress through this assembly line and end up out the other end as undigested material. So those people that are lactose intolerant take in, right, milk product, right? And they get like stuff up the cramp, and sometimes they get diarrhea and stuff like that because these materials are going through without being absorbed. Okay. So we have one for carbohydrates, one for proteins or peptides, and then we also have intestinal lipase. So we're probably getting down here, breaking down the food items to small units that we can absorb. So we even have these embedded digestive enzymes in the wall of small intestine. Okay. So the same type of control mechanism that we've seen before, we're also going to work for the activity of the small intestine, for instance, right? Parasympathetic nervous system is going to stimulate the small intestine, increase secretions, etc. Okay, so now we finally got down to the point where we broke down the food we fit in small units that are going to hold it across the body. So that's the primary role of like the latter part of the jejunum and ileum. We migrate, march all through the small intestine, we're breaking it down, putting more enzymes onto it, etc. And now we're going to be pulling it across the body. So the major type of absorption for both food fuel molecules, like I said, like carbohydrate products and protein products, etc., and water. Because we tend to forget about the water. Water is very critical in keeping our food balanced. All right, so what we usually do then in the small intestine is we've got mixing type movements, right? And the mixing type movements that are in the small intestine is actually called segmentation. And so segmentation is where we're going to have a mixing type of back and forth, a little bit like something, right? But we just like kneading dough, where you squeeze it in the middle, and the pinches is separated this way. When you pinch it out of there, that pinches is separated that way. So it's mixing it and it comes apart, etc. So there's going to be a big movement back and forth movements of the small intestine. And there's also going to be those Cheroske waves, the rhythmic contractions that are going to propel the food further down their alimentary canal. Okay? So after that, we get to what's called the ileocecal junction, the ileocecal sphincter. So if you look at ileo, that reminds us of ileum, the last section of the small intestine, and cecal, that was like cecal a little bit, and that reminds us of what we have when we get to the larger intestine. But the very first pocket of the larger intestine is called cecum. So you see this pocket right here. That's called cecum. So the ileocecal valve is going to control the movement of food from the small intestine to the larger intestine. Now, when I say movement of food, that's not entirely true because we just said that all the time and effort breaking down the food so we can absorb from the small intestine. So by the time we leave the small intestine, most of our food and nutrients, as well as water, should have already been absorbed. So what rotting here is the cecum, and what's going to go around this large intestine is going to largely be undigestible food items, okay? So for instance, when we're talking about things like fiber before, right? Fiber supposed to move through your system well, right? That's because it's a plain carbohydrate that starts your enzymes to work very well. Now, when you chew up corn, right? The better that's going to be digested to a certain extent. But we all know that corn can go through pretty much any fat, right? It's got a lot of these starches in it. So anyway, by the time we get to the larger intestine, almost basically all of our nutrient absorption is already occurred. Okay? So the only thing that the larger intestine is doing is storing some digestible material, the feces, and it's absorbing the maximum amount of water and electrolytes. The water electrolytes in. It's like a picnic dust we throw in the catering, stuff like that. That's magic stuff, right? So they're very salt, right? Electrolytes go down to the electrical current. They're charged particles, ions, sodium ions, potassium, sodium chloride, etc. Okay? So the large intestine will absorb a little bit of water and a little bit of electrolytes, but it's really good cleaning food. Cleaning food is like a drying rack for this undigestible food. Because along the way, we've been trying to go from nutrients and fluid out. Remember when we were taking something? It was a thick soup. And now, in the small intestine, we can pull and digest and pulling stuff out of it. So by the time we get to the larger intestine, it's becoming semi-solid instead. And so when we look at the large intestine, we also see structural adaptation that helps it meet that need of moving that metabolic material. So we don't have anymore. We don't need all that surface area anymore, but we do notice that the muscle that we have there is a lot bigger, right? Because the tougher to move this metabolic material. So you can see it kind of looks like a caterpillar. It's got little legs to it, and we have smooth muscle bands that kind of band around and creating these little pockets that we call posture. Okay? But every place where it pushes in, we've got a stronger smooth muscle band that are going through around it. And you see that little as well, that is like the coli. Those are large, longitudinal muscle bands that will again help you squeeze it down. Okay? So there are a few different sections of large intestine. Large intestine is also known as the colon. It's basically the same thing. Okay? So we start out with the think of it in that lower right-hand corner. Okay? So that's where the small intestine comes into the large intestine. So we start in that lower right-hand corner, lower right-hand corner. We have ascending colon, ascending large intestine. Bacteria reverse, going across. A descending going down the left side. And then see how it's an S shape right here? So that's called sigmoid colon. And then straight out that comes the rectum. And then it comes out the last segment right before the anus, the smooth muscle band, as well as the skeletal muscle band that controls movement out of it. Okay? And the appendix is a kind of relic, lymphatic structure, right? That's a bit like mom, if you will, where we've got resident populations of cells from white blood cells that are inside it, but it takes off of the cecum itself. And so if you think they have appendicitis or something like that, it's better be on the right side. On the left side, it's just gas. Okay? Okay. So like I said, large intestine is basically a big ride. It doesn't digest or absorb nutrients. It doesn't secrete mucus, like I said, every section is going to, and it will, as we know through in a dry tract, absorb some electrolytes and some water. Now, it does contain symbiotic bacteria, symbiotic E. coli, things like that. In fact, there's enough bacteria that's actually coming out on bacteria in your body, on the surface of your body, and like opening of your body, especially in the large intestine. There are so many bacterial cells there, and these are beneficial bacterial cells, not pathogenic ones. But they think there are more bacterial cells associated with your body than there are cells of your body. There's that many of them. They're really small compared to our cells. And a lot of them, like I said, are going to be beneficial ones that are taken from the large intestine. And those bacteria, you say they're symbiotic, they're beneficial to us, we can count them. When they get to us, they will synthesize some vitamins for us, like vitamin K that you hear about, and they can also help break down cellulose, that plant starch. Our enzymes can't break that down, but the bacterial enzymes can. So fermentation, stuff like that, can break down that cellulose. Now, does it do as much good? Not a lot of good. In fact, when you eat high fiber meals, stuff like that, like beans and things like that, then those bacteria produce things like methane gas, or the whole lactulin thing, comes bacteria breaking down those plant starches. So, pros and cons of bacteria. Okay. Well, we are just about to the end now. What we're moving next is going to be the nutrition section, where we're looking at the different molecules that we're breaking down, like carbohydrates and lipids and proteins, and just kind of reminding ourselves of those, talking about vitamins and other types of things that we need to get from our diet. So it's not